everyone. I'd like to welcome those of you in the room and those watching online to the American Society for Microbiology's Halloween edition of Microbes After Hours. I'm Catherine Lontock. I am the Public Outreach Manager here at ASM, and I'd like to introduce our guest speaker for this evening, Dr. Jennifer DeBruyn. She earned a Bachelor's of Science with honors in biology at Queen's University in Ontario. Then she went on to receive a PhD in ecology and evolutionary biology at the University of Tennessee. And she is currently an associate professor in the Department of Biosystems Engineering and Soil Science at the University of Tennessee. Her research focuses on microbial ecology. And tonight, we're going to hear specifically about some of the work that her lab has been doing on microbial communities that decompose dead bodies. So after the talk, we will have Q&A. For those of you in the room, you can line up at the microphone right there. For those of you online, you can submit your questions through the YouTube chat or on Twitter using the hashtag ASMLive. All right.
Thank you very much, Catherine. Thank you for having me here tonight, and it's a real pleasure for me to be here and tell you about some of our research on the necrobiome, or the microbial communities that are decomposing dead carcasses and cadavers. I'd actually like to start by having you think about living animals and their ecosystems, because this is typically how we think of animals and their interactions with the environment. We think about what they eat or who eats them, their role in the food chain. We might think about how they're contributing to diversity. And we might think about modifications they make to their environment, such as this industrious beaver building his dam. But we think much less about what happens to those animals after they die. But they're still part of the ecosystem. So this dead beaver here has been accumulating tissues and carbon and nutrients all his life. And after he's gone, all of those nutrients and carbon needs to be recycled back into the environment. And so this is still very much part of the ecosystem and a really critical process. If this wasn't going on all this time, this redistribution and recycling of nutrients, we would have a planet covered in dead plants and animals. So what are the organisms that are involved in recycling a dead carcass? Well, we get a whole suite of organisms that move in to do this recycling. Some of them include the scavengers, so these could be avian or mammalian scavengers that will come in and feed on the tissues before they're decayed. We have the microbial decomposers, so these are the bacteria and fungi that are dissolving the tissues and mineralize them back to their constituents. And this is primarily the groups that I work on, that I'm studying, and I'll be telling you a little bit more about tonight. We also have a number of insects that take advantage of a newly dead animal. There's a number of flies that will lay either their eggs or their larvae in dead tissue, allowing those larvae to digest the tissue and gain energy before they migrate away to pupate later in life, so they're gaining energy from the system. There's also a number of insects that directly feed on the tissue, such as carrion beetles. In addition to the primary consumers of dead tissues, we also have all these secondary consumers that move into the systems. So because of this big bloom of bacteria and fungi all over the dead carcass, we'll get organisms that feed on bacteria moving into the system, such as nematodes and slime molds, and they just come in and kind of scoop through all that nice bacterial bloom. Likewise, there's also a number of organisms that take advantage of fly larvae as a delicious nutrient source. So these are things like moths and wasps, predatory insects. Some of them will actually lay their eggs into maggots and larvae to develop there, or they'll feed on them. It's not common for us to see butterflies and birds visiting and picking off maggots as they decompose. So this one animal contributes to this whole food web of organisms, this whole little pop-up pocket of diversity in an ecosystem. And I think it's a realm of diversity that we just don't often think about for the ecosystem. And one of the reasons is because it is very rapid and very ephemeral. It's a transient system. It's just here for a short period of time. All these nutrients are fairly rapidly remineralized and cycled back into the environment, and in biogeochemistry, we refer to this as a hotspot. It's something that has a great deal of cycling and activity compared to the surrounding environment. We also call these things hot moments, meaning that it's a transient or ephemeral in nature. It's just decomposing for a very short period of time and then it's gone. You go for a walk in the woods, you see lots of dead leaves, you don't see a lot of dead animals, and that's typically because they're consumed so rapidly. And one of the reasons that it's such a rapid and transient event is just because of the nature of animal tissue. So we think about how animals, what they're comprised of compared to plant materials. Plant materials have a lot more carbon in them and will decay much slower. So if we look at a typical decomposition curve for a plant measured as mass loss over time, we see that plant materials will decay at a fairly steady and rapid rate. An animal carcass, in contrast, will decay very rapidly early on and exhibits these really steep mass loss curves. And that's because they are so moisture and nutrient rich compared to plants. So, for example, you take this tree here, obviously has a lot more biomass than the feral pig down below, but they actually have about the same amount of nitrogen in them. So it's a really concentrated pocket of fertilizer is one way to look at it. And so to show you what one of these looks like in person, this is a beaver carcass that we were experimenting with this summer. And I hope it shows up on the picture, but you can see this is about 6 days of decomposition. And you can see that it is no longer a flesh and bones organism. And we see this big site of decomposition fluids where the soils and the surrounding areas are darkened with all the nutrient-rich goo seeping in. So it forms this visible hotspot. And so what's going on in these hotspots? Well, like I said, there's a lot of remineralization of compounds, and this is primarily done by the decomposers, the bacteria and the fungi. And we see lots of evidence of their activity in these systems. First of all, we typically see elevated temperatures. That shows us that microbes are working away. And we see a decrease in oxygen. Again, these microbes are consuming oxygen as they're decomposing. You get an oxygen drawdown, and you tend to get anoxic conditions forming rapidly. In the soils below, we see big changes. We see increases in pH. We see changes in electrical conductivity, which is a measure of the salts in the soil. We very typically see increases in ammonia, which is one of the key decomposition products of proteins. And it's not just the soils below, but we also see off-gassing of a number of compounds that show us that microbes are hard at work. We see increases of CO2, which is a general metric of microbial respiration. So this is just showing, again, that these microbes are very, very active. We see increases in nitrous oxide, showing us that denitrification is likely occurring. And we also see increases in methane, showing that methanogenesis may be occurring. So what this tells us is that there's a vast diversity of microbial metabolisms at play. It's not just one type of decomposition pathways, but we got a whole diversity of taxa and doing a whole diversity of decomposition, and that's just because of the complexity of the system. So before I get into some more of our specific research, I'd actually like to tell you a little bit more about some of the areas that are interested in this type of research. Obviously, as an ecologist, I find this a fascinating system, how nutrients are being redistributed in our environment. And you can think about scenarios maybe where you have changes in animal migration patterns or mass animal die-offs, where this could really change where nutrients are flowing in our environment. But there's a number of other groups of people, of scientists and practitioners that are also interested in decomposition research, and so I wanted to highlight some of those areas as well. The formal name for the study of decomposition is taphonomy, and taphonomy concerns itself with understanding how the environment affects a decomposing carcass and how the decomposing carcass affects its environment. So from a long-term preservation standpoint, from a fossilization, looking at our fossil and biological record on the planet, this is really interesting to understand what conditions were present in order to contribute to the formation of that fossil. obviously that can tell us a lot about historical conditions on our planet. And so geologists have long been using taphonomy as a formal field of study. More recently, forensic science has adopted taphonomy as a field of study. And forensic science is very interested in how we can understand processes and patterns of decomposition to give us information about a decomposing body. So they're very interested in human decomposition processes. And so they will be interested perhaps in using rates and patterns of decay to tell us approximately how long a body has been decomposing. That can help them pin down the postmortem interval and hopefully come up with a window of time in which that person was killed. So that's a very important piece of information in a lot of criminal cases. They also may use patterns of decomposition to tell them something about the manner of death. So for example, we know that if a particular body site had some trauma or an open wound, those parts will decompose faster than the rest of the body. So if you find a partially decomposed body where there's differential decay on different parts, it can suggest that there may have been some sort of trauma or wound to a certain part of the body. It may tell you something about the manner of death. Another way that forensic science likes to understand decomposition is in looking at the products of decomposition, and particularly some of these metabolites and nutrients and the changes in the soil chemistry and changes in plant chemistry to help them find hidden or clandestine graves. So is this fertilization event and enrichment, a lot of times we'll see excessive plant growth in the area of a grave. And there's been some evidence now that those plants are actually enriched in certain nitrogen isotopes. And can we use some of those signatures to help find a hidden grave? And so those are some of the questions that forensic science is hoping decomposition and a better understanding of postmortem processes can help them answer. And so there's a number of facilities now that exist that are devoted to the study of forensic taphonomy. These are very popularly known as body farms, but that's not the formal name for them. And one of the longest running, the first one that was established, is actually the University of Tennessee Anthropology Research Facility. This particular laboratory, it's a 3-acre outdoor laboratory. It's been running for 36 years now. It was established by Dr. Bill Bass in 1980. He was the state forensic anthropologist at the time. And he realized that at the time, when it came to estimating how long someone had been dead, estimating postmortem intervals, that they didn't have any empirical evidence to back up their estimations. It was all just based on a case-by-case study, and they needed a way to robustly test, replicate, and experimentally confirm these patterns of decay. So he went to the University of Tennessee administration at the time, and this was the late 70s, so they didn't give him much pushback, and they said, sure, you can have a couple acres of land and some cadavers, and that's how the Anthropology Research Facility was born. And it's been at the forefront of a lot of forensic taphonomy research. It's really been an exciting place to collaborate and work with because they have been involved in so many of these studies. They look at patterns and processes of decay under just about every conceivable scenario you could imagine, all generally inspired by real criminal cases. So they look at clothed versus unclothed bodies, indoors versus outdoors versus trunk of the car, hanging from a noose, you name it, and they've probably studied it at the facility. And so this has brought a lot of experimental, empirical evidence to the field of forensic science and forensic taphonomy that's been really, really important. So it's been very exciting to work there. And one of the reasons this particular facility is so successful is they maintain a very large body donation program. So about 4,000, more than 4,000 people are actually pre-registered to donate their bodies to this facility. So it is just a really special and amazing place to work. And so they've worked at this facility now for 36 years answering these questions about decomposition, but more recently attention has turned to microbes, and if we could use microbes as some sort of indicator in forensic cases. And so this idea really is inspired by forensic entomology. So forensic entomology, if you're not familiar with it as a field, essentially uses the known growth rates of certain species of fly larvae to back calculate how long it's been since that egg was originally laid. So they know from a myriad of lab studies exactly how long it takes a blowfly larvae to reach a certain size. So if you collect a maggot off of a decomposing body and you know the ambient temperature, you can back calculate when that egg was first laid. So this has been a robustly evaluated field. It is not without its variation and without some challenges, but it is it has been used as evidence in criminal cases, so it's a valid field. And so the idea is maybe we could use patterns and processes of microbes in these systems to estimate postmortem interval. One of the big advantages of using microbes is that we don't have to worry about colonization. Microbes are already in us and on us and all around us, so we don't have to worry about flies having access to the body. Forensic entomology doesn't work where flies can't get to the body, either because it's wrapped in plastic, or it's buried, or it's winter and there's just not a lot of fly activity. So there may be some advantages to employing microbial approaches. But in order for this to be successful, we really need to have developed a database of robust patterns of microbial succession. And we need to know how universal these patterns are. So are the patterns that I'm observing in East Tennessee the same patterns that people are seeing in Texas or Alaska or Hawaii or anywhere else? And so we're still at the very, very early phases. This is still more of a kind of cool idea. Let's start documenting and see if this is valid. So it'll be a while, I think, before we see if this becomes a legitimate forensic tool. But it is certainly fun to think about. One of the other applications of decomposition research is in animal mortality management or livestock mortality management. So animal carcass waste is a byproduct of our system, either from livestock farms or nuisance animal populations like these feral pigs up here. And so one of the things my group is doing is we're experimenting with best practices and different materials for composting animals to get rid of them quickly and cleanly, preventing the spread of pathogens and preventing scavengers and odors and other nuisance things. So this is an experiment here. We ran a couple years ago with feral hogs, and we found that working with about 3 feet of mixed wood chips in this system, we could get temperatures high enough to kill off pathogens, and the entire pig, bones and all, was gone in about 3 to 4 months over the summer. So managing these things well can actually be quite useful. In a similar fashion, people are starting to think about these ideas for human decomposition as well, and there's been a movement towards green burials. People are getting a little bit fed up with traditional funeral practices such as embalming and caskets and maybe some of the more toxic and less environmentally friendly approaches. And so people are interested in having less of an environmental impact after they die, and they're exploring more sustainable or more natural options. And essentially all these are options that take advantage of decomposition and promote decomposition instead of trying to stave it off as long as possible. So some examples of this are biodegradable caskets, for example, these ones made of bamboo here. Some of the more elaborate options out there is this mushroom suit. If you haven't seen it, there's a really interesting TED Talk on this particular suit. It's called the Infinity Burial Suit, and it's a biodegradable cotton shroud that's impregnated with fungal mycelium spores. And the idea is that it would kind of rapidly decompose once you're wrapped into it and just add water. Kind of reminds me of the chia pets. There's a couple more conceptual ideas out there that are interesting. I pulled out these Capsula Mundi pods. This is an Italian company that's designed, I don't think they're producing, but they've designed these biodegradable starch pods that you would put the body into, and then a tree would grow at the top, basically converting yourself into a tree. And then one of the more interesting ones that I'm personally a fan of is the Urban Death Project, which is the brainchild of architect Katrina Spade. And this is just in the design phase right now, but she's designed a human composting facility. So taking the ideas, the things that we're doing with livestock composting, and asking if we can use this for disposal of humans in a green and efficient manner. So she's designed this facility that maintains the solemnity and the ritual of a graveyard, but is essentially a compost pile that you could lay your loved ones in and get a pot of soil back 2 to 3 weeks later. So I think it's a very interesting idea. Another application of decomposition research that you may not have thought of, and I certainly hadn't up until a couple months ago, is the forensic restoration industry. So these are crime scene cleaners, if you will. These are the people that will go in after a murder or a suicide or an unintended death, and they have to clean up the house or the building and restore it to its natural state. I was just at a forensic restoration conference last week. I had the pleasure of meeting with some of these specialists and talking with them about their work and what their biggest concerns are, and they're very interested in postmortem microbes from a pathogen perspective. They want to know what kinds of risks they have. If that person had an infectious disease, how likely are those pathogens going to persist in the decomposition environment? And what does this mean for site worker risk? So hopefully that gives you some ideas of some of the people that actually care about decomposition above and beyond it just being a cool and interesting process. So next I'd like to tell you about some of the research that my lab is doing into the microbes that are in this system. So I'm going to get a little bit more specific on the decomposition processes, and I would also like to say at this point that I will be showing a few pictures of decomposing human cadavers, so just as fair warning, if you find that disturbing and want to look away, that's perfectly fine and normal. It won't be too bad. And it's a small screen. All right, so after an animal or a human dies, what happens is the heart stops pumping blood, so you stop circulating blood and you stop breathing. So oxygen is cut off to the cells. When that happens, the cells start shutting down. They undergo a process called autolysis, which is essentially self-digestion. They release these enzymes called lysozymes, which start chewing up the cells and breaking them down into their macromolecules. It's cell suicide, if you will. And so your cells, as they're breaking down and releasing all these carbohydrates and proteins and fats, there's now a whole bunch of food for your resident microbes to take care of. So thanks to the human microbiome and some of this research on the microbiome, we know a lot about our resident microbes, and we know how important they are in our health while we're alive. After we die, however, they're not so friendly to us, and they essentially start digesting us from the inside out. So the first stages of putrefaction, or this microbial conversion of cellular products, is primarily carried out by our microbes, and particularly the microbes in the guts, because it's such a dense population of microbes. And so they'll be converting these macromolecules into constituents. Now, there's a whole suite of compounds that are produced, but I've tried to summarize it here into a few main categories just to give you an idea of just how complex decomposition fluid, decomposition goo is. So carbohydrates will be converted to organic acids, and if there's oxygen present, we actually get production of CO2 and water, which is the cleanest and least smelly way to decompose everything. This is what you're going for in your compost pile, is oxygenic decomposition. In our bodies, however, in the body cavity, there's no oxygen, and so fermentation predominates. And so we get a production of organic acids, alcohols, this is your classic fermentation pathway, methane, and hydrogen gas. Proteins will be broken down into their constituent amino acids, and those will be further converted into carboxylic acids, polyamines, such as cadaverine and putrescine, and you can guess from the name of those that those are the compounds that give decaying flesh that signature smell, if you've ever smelled it. Polysulfides, also very smelly, and aromatics. Indol, scatol, and phenol are very common. You may have smelled phenol because we use it as a solvent. Scatol is what gives feces its signature smell. We also get production of ammonia gas, which is quite smelly. Hydrogen sulfide is the smell of rotting eggs, methane, and carbon dioxide. Fats will be broken down into fatty acids and glycerol, and in the presence of oxygen, we can get conversion into aldehydes and ketones. Again, these are both very smelly compounds. Aldehydes are frequently used in perfumes. Formaldehyde might be the one that you're most familiar with. Ketones, a common example of that is acetone, which is a frequent solvent, nail polish remover, so very, very aromatic compounds, very smelly compounds. Without oxygen, fatty acids will actually be saturated and can be converted into adipocere, or something known as grave wax. This is kind of like a fluffy lard-like substance that's not uncommon to find in burials or places where you have decomposing tissues without oxygen. So just an overview of the suite of compounds that are in the system and coming in, the products of microbial decomposition, but then also become fodder for the next suite of microbes. One thing I'd like you to appreciate is how many of these products are gaseous in nature. So with all these gases building up as a result of fermentation, we end up getting bloating of the body. And so a very normal stage of decomposition is to see this distension of particularly the torso and the face and the groin areas as the carcass or the body will blow up with all these gases. And of course, at some point, those gases have to find their way out. And so it will either rupture the body, or they'll find their way out through existing orifices, or the whole thing just disintegrates. And we enter a period of active decay. Now, active decay describes the period when we have the most rapid mass loss of the body. So I showed that curve earlier where you have this kind of sudden decrease in mass. That's active decay. We've got all the gases escaping. We have a lot of fluids leaking to the soil below here. Hopefully you can see a very defined fertility island where those decomposition fluids are leaking in all around the body. And we will have oxygen, and external microbes are introduced into the body, and so there will be very rapid oxygenic or oxygen-dependent decomposition of the body at this time. To give you an example from East Tennessee, from my part of the world, a 150-pound male can be reduced to about 30 pounds of tissue in about 2 days over the summer during this process. So it happens very, very quickly. We get a loss of all that. You also see that this is also the time when maggots can be most active. So the gray parts that you see are all maggots. And as evidence of how active this time is, if we look at internal temperatures of this cadaver, we see elevated temperatures during this period of active decay. So just showing that it's not just fluid loss, but there's a lot of microbial activity at this time. And then when most of the soft tissues are gone, we enter a period called advanced decay. There are still tissues present on the body, but it tends to be the more recalcitrant compounds, some of the skin, keratin, hair, and those sorts of harder to digest compounds. Decomposition will still happen, but much more slowly. Temperatures drop down to ambient, and we see a drop-off of oxygen at this time. So in the system, we really have three main microbial communities at play. I've already talked to you about the gut microbes, your resident microbes, and they're the ones that are active in decomposition from day one. They're converting all those cellular products, and so they're quite busy, and they'll eventually get flushed into the soil along with all the decomposition fluid once they go down. The second community we have to consider is the resident soil microbes. So these are microbes that are just normally present in soil and now have received this flush of nutrient-rich goo into their system. And so microbes that are able to take advantage of this, that grow quickly and can bloom under these scenarios and maybe aren't too sensitive to the high ammonia content, will actually do very well. So we'll get a bloom of these opportunist soil microbes that join in. The third party to consider is all the visitors to the cadaver. So at the beginning, I showed you the whole ecosystem, the network of different organisms that will come to feed off of this cadaver. And so they, of course, all have their own commensal microbes. And so some of those may be getting into the system as well, particularly when you think about how much of a carcass might be covered in maggots, for example. That's a huge influx of maggots and their associated microbes. And so with all the changing chemistries and with these three populations, we actually do see big shifts in the microbial communities in the system. So for example, this is our data on microbial communities in soils below decaying human cadavers. And for reference, this first bar here is the gut microbial communities of the cadavers that we used. And it looks like any other typical gut community. We have Firmicutes and Bacteroides as the dominant players here. Right next to it, the next two bars are soils that haven't been impacted by decomposition. So they are typical of what you would expect a soil to look like. They have a good mix of taxa, including proteobacteria, acidobacteria, verrucomicrobia. And then as decomposition progresses, my arrow keeps disappearing, there we go. As decomposition progresses through the bloat and then through the active, this is when most of the fluids are hitting the soil, and then on through advanced, we see that there's a big shift in microbial communities. One of the most noticeable ones is in Firmicutes, which is this light blue at the bottom, I hope you can see. Firmicutes goes from being very rare in normal soils to over half of our community by advanced decay. So this is a massive swing in the types of microbes that are in the system. We also see disappearance of other groups. So for example, the acidobacteria, which make up a good portion of our normal soil microflora, almost entirely disappear in decomposition soils, either because they're being outcompeted or they're just sensitive to the high ammonia levels or other toxins in the system. And the reason these changes are occurring is because we have this positive feedback going on. What's happening is we get the decomposition by the host microbes that are releasing these compounds, these metabolites and decomposition products into the soil. That's selecting for soil microbial communities that can live and eat these compounds. And those microbial communities then participate in decomposition further. So you end up getting this positive feedback cycle that's causing this succession of microbial communities over time. And it's this succession that the people who are interested in maybe using microbial communities as forensic indicators are starting to look at. Are these successional patterns and processes consistent and robust enough that we could actually use them in some sort of evidentiary way? If we look a little bit closer at some of the taxa that are most significantly changing in grave soils, we can learn a little bit more about who they are and what they might be doing. So for example, this heat map shows the abundance of different taxa by the time during decay, with blues being the highest abundances. So we can see that some groups, like our acidobacteria and our verrucomicrobia, are high early on and decline to almost nothing later in decay. We also have a couple other taxa that show up in quite high abundances about midway through decay. So they're basically not present at the start, and they appear in our decomposition soils. And if we look more closely at what some of these taxa are, we notice that a lot of them were anaerobic taxa. So these are either obligate or facultative anaerobes that are sensitive to high oxygen. And so this told us that we're shifting the chemistry of the system such that we're actually moving it to an anoxic system where these anaerobes can proliferate. Normally they wouldn't be able to compete. We also notice in our communities that we're finding insect-associated bacteria. So here's just 3 examples, but we're noticing these taxa related to other things that have been isolated either from flies or fly larvae. So that sort of confirms our idea that these insects and visitors to the carcass are also bringing microbial communities into the system. We also looked at human-associated bacteria in decomposition soils. So I told you how active our gut microbes were early on. And so we wanted to look specifically at some of those taxa and see if they are persisting and playing a role in the soils. So what this is a list of bacterial taxa that were present in the guts of our cadavers, shown here, but absent or very, very rare in soils. And what we looked for is whether they appeared during decay. And we found that several of them indeed did show up in soils as decay was progressing. And that wasn't very surprising to us. That's what we predicted. They would get flushed into the soil along with all the decomposition fluid. What was surprising, though, is how long they persisted. we figured that they wouldn't have a fighting chance out in the soil. You know, a gut microbe is adapted to 38 degrees Celsius, no oxygen, a steady flow of carbohydrates. It's a pretty comfy life inside your gut. Once they get out into the soil, it's kind of the Wild West. You have changing environmental conditions, you have soil microbes that are much better adapted to that environment. We thought that they would be outcompeted and they just wouldn't persist. So we're actually surprised to find that some of them lasted throughout the whole decomposition study. One in particular that we tracked was Bacteroides, which is an obligate anaerobe, and found that it actually persisted in high levels in our decomposition soils up to almost a year post-mortem. So this was much, much longer than we originally thought. And it brings up questions about persistence of pathogens in this environment as well, which we thought shouldn't last, but maybe they are. And what we think is happening is in this environment, the gooiness of the decomposition fluids, for lack of a better word, is really limiting oxygen flow into the soils, and we're maintaining anaerobic conditions for long periods of time, and that's allowing some of these anaerobes to persist. So we're starting to put together a picture of what our decomposer communities look like, and we're combining data from biogeochemistry, which I showed at the very start of the talk, some targeted metagenomic approaches where we specifically look for taxa, as well as shotgun metagenomic approaches where we look for functional genes as well to try and get a picture of what these organisms are doing. And so what we've pieced together so far is that we have these early decay communities that are what I'm calling first flush responders. So these are things that respond to this fertilization event quickly. They consume oxygen rapidly. They're involved in that first step of decay. They use the easy stuff. We can think of them kind of the weeds of the system. Later in decay, though, we get a community that has a much lower efficiency, indicative of a lot more anaerobic metabolisms. We see more anaerobic taxa, such as Firmicutes and other things. We see human and insect taxa come into play later on in decay. And we're also starting to gather evidence of anaerobic respiration genes, stress response, and pathogenesis and defense system, genetic systems. So it's showing us that these communities are not only changing in terms of what they're doing, they're changing in terms of the types of microbes that are there and the different functions that they can perform. So we're getting a much better picture of how the ecology of this system. So another thing we wanted to know is how universal this response was. So we had an opportunity to collaborate on a project where they were comparing pig and human decomposition. So pigs are frequently used in forensic taphonomy research as a human analog. So in cases where you don't have anthropology research facility, you don't have access to human cadavers, a lot of people are using pigs. They're probably the closest thing in the animal world to us. They don't have fur, they have hair, they're about the same size as us, and we're both omnivores, so we think we have relatively similar gut communities compared to other things. And so we went into this and we thought that humans and pigs should have pretty similar responses in terms of decomposition. So looking at respiration, or just a general measure of microbial activity, we see that both humans and pigs decompose. There was a lot of microbial activity, as we had expected, a little bit higher in humans, but they seemed not too far off in that sense. What surprises us, though, is that we saw differences in the soil below the pigs and the humans. One thing we noted was that humans, the pH actually dropped off quite dramatically. Now, if you remember all the way back to the beginning of the talk, I showed a graph of pH where it went up. And this is something that seems to vary based on the study. In some cases, pH is quite elevated, and in some cases it goes down, and I don't know that we've quite figured out why. But in this case, humans had a drop in pH, whereas pigs had slightly elevated, though not significant pH, shown in blue here. And then when we look at specific microbial activities, in this case we're looking at leucine aminopeptidase activity, which is a marker for protein decomposition activity, we saw that there was no elevated activity in humans, but we saw elevated activity in pigs. So we don't know why this is happening, but it certainly speaks to the fact that humans and pigs may not be as similar as we thought, and that some of these decomposition processes may not be as universal as they thought. So it may be dependent on the host composition, on the muscle to fat ratio. We're not really sure, but these are interesting results that we want to pursue. Another thing that we're working on right now is looking not just at the microbial communities, but also at the metabolites that they're producing. So as microbes are degrading, they're creating all these small molecules. And so we can use an approach called metabolomics to get at some of those compounds. So this is an untargeted approach. We're not looking for anything in particular. We essentially run the entire sample on a GC and look for all the small water-soluble metabolites in the system. We compare it to a library to try and identify what metabolites are there. And the idea is here is that these metabolites will tell us what microbial pathways are being used, and maybe they could be used as signatures of decomposition soil. So there may be a forensic application for these as well if we know what markers of grave soils are present. And so these results are still early, but we're starting to pull out some patterns. For example, we noticed that some compounds are higher, which is indicated in orange here, earlier in decomposition. Some things are high throughout, and other things show up late in decomposition. Not surprisingly, a lot of things that are being elevated during decomposition are amino acids and other protein degradation products, and carboxylic acids, and some of the things I alluded to earlier. So we're seeing some of these signatures in grave soil. So we're just in the process now of sorting through this data and trying to document some of these patterns in a more statistically robust way. And then I'd just like to end tonight by talking about our newest project on post-mortem microbial communities, and that is dealing with the skeleton microbiome. I'm collaborating for this project with a forensic anthropologist that's very interested in using skeletal remains for identification purposes. And one thing that she's noticed is that different skeletal elements, so different bones, actually give you different yields of DNA. So we get different amounts of human DNA out of these bones, and their quality differs by element as well. So some of them, they can get full STR profiles and robustly identify the person, and other elements are not working so well. And because it's quite laborious and expensive to drill into bone to extract DNA from, they want to get a better idea of what skeletal elements are best to get DNA from for identification purposes. So noticing the variability in human DNA recovery from these got us to thinking about perhaps a microbial degradation element to the story. So maybe one of the reasons that we're not getting high quality DNA out of some of these elements is because microbes have invaded and they're degrading the DNA, or they're just co-extracting so much microbial DNA that their assays aren't working. So what we're doing is exploring the skeleton microbiome, or the microbial communities that have invaded and are living in bone. And one thing we've noticed is that just like the human DNA results, we're getting different amounts of bacterial DNA out of these elements, out of different elements. So it shows us that there's different amounts of bacterial pressure in these bones. And then our first pass at looking at the composition of these microbial communities, we actually see that the types of microbes are different by skeletal element as well. So in particular, we're seeing that microbial communities associated with the hands and with the teeth appear to be unique compared to other bones. So why this is, we don't know at this point, but we're really excited to start in on this project and get some answers to that and see how these microbial community parameters possibly relate back to human DNA preservation. So I would just like to take a moment now to thank the families of the donors of the Bass Donated Skeleton Collection. It is incredibly generous of people to donate their body or their loved one's body to the program, but without it, none of this research would be possible, so I am very, very grateful to them always. The research I showed you tonight was carried out by members of my lab, as well as faculty and staff at the UT Forensic Anthropology Center. We collaborate with the UT Mass Spec Corps on the Metabolomics project, and we also have a couple other collaborators both at UT and Bodie. This work has been funded by the National Science Foundation and the National Institute of Justice. So thank you.
All right, so we'll take questions now at the microphone. If you're online, Again, you can enter your question into the YouTube chat, or you can post it on Twitter with the hashtag ASMLive.
So I have a question from the YouTube chat. What is the most disputed or perplexing evidence of prehistoric decay that doesn't quite correlate to today's biome?
I don't know. I don't do old decay. Ask a geologist. I don't know.
So I'm wondering if your studies are typically on a single cadaver or carcass, and often there are things like wars or mass graves or fish kills, for example. And I'm wondering if the processes that occur under a single body that's decaying are the same when you get a massive amount of material.
That's an excellent question, and I'm not aware of any research right now that has compared single to a mass die-off event for humans anyways. We do at the Forensic Anthropology Center, we do currently have a mass grave project ongoing. And so we're hoping to collect soil samples from that and start to examine some of that. Trying to think of if there's animal equivalents, but nothing comes to mind per se. Yeah, fish kills would be the big one. So there's some work on salmon and massive die-offs of salmon and how that transports nutrients from one place, essentially the ocean, upstream and river and some of those, but yeah, good question.
So I was wondering how our diets might affect, I mean, you mentioned something about the gut bacteria. Are there big differences that you see in decomposition, for example, in people who are vegetarian or vegan versus omnivore? And also, are there differences in just the composition of our bodies?
That's a great question. So to start, we do know that even though our gut microbiome does vary by diet, it's still quite drastically different than, say, a soil community, okay? So we do see a lot of commonalities in gut microbiomes. But in our work with these cadavers so far, we see a huge amount of variability in our data that is unexplained right now. We just know that individual variability is quite high. And this came out most noticeably during that human-pig comparison study. So we had 5 humans and 5 pigs. And in general, in most of the things we measured, the error bars on the humans are way bigger than on the pigs. The pigs were just much more consistent in decomposition processes. And so when we're thinking about this, we realize, well, the pigs are all the same age, they came from the same farm, they've been fed the same diet. So they're probably very similar in composition. Whereas the humans, we take the donors that we get, and we don't always know that much about their diet or lifestyle. And so we think that some of those diet lifestyle factors may play into it. The other thing we wonder about is medication. So if these people are on antibiotics before they die, we know that has huge impacts on your microflora, and so that could change decomposition. But we just haven't had the study sizes yet to be able to tease out those factors yet. But it's definitely something we'd like to explore.
You mentioned something about the plant life that incorporates some of the elements from the bodies afterwards. Mm-hmm. I'm wondering if I can imagine that maybe some of the soil or the microbial hallmarks of decay might dissipate kind of rapidly. But if you have a tree or plant life, how long does that serve as sort of evidence that maybe there's a grave or something happened there?
Also a very good question. So this area of research was kind of more anecdotal at the start, where you would see a carcass and you'd see kind of more plant growth kind of in the immediate area of it. So it seemed it was enriching for for nitrogen. I am aware of one study that looked at muskox carcass in the Arctic, and they actually found that the plants surrounding that carcass 10 years later were still elevated in nitrogen from that one die-off event. So I don't know if that's just a tundra-specific response. I don't know how that translates to smaller animals, but it certainly suggests that these impacts can be long-term. Good question.
Someone asked, do the differences in the skeleton biome reflect differences in physiological makeup, such as does relative muscle density of a body type or relative bone densities affect the different organisms found?
So one of our hypotheses going into this bone project is that the bone density is what's influencing the recovery and the microbial invasion. So you have more dense cortical bones. These are your weight-bearing, like your femur bones. And then you have the more honeycomb cancellous bones. These are more open structure bones, and those will be some of the smaller bones. And so one of our hypotheses, still to be tested, is that the more open structure of the cancellous bones allows microbes to get in and chew through some of the organics much more easily than the denser cortical bones. So we think that there's a physical component to this microbial invasion, but that's very much just a hypothesis at this point.
What attracted you to this specialty area of microbiology, and why did Tennessee become this center of research in this area?
I will start with the latter question first. So the reason that Tennessee became the center for this study was really due to Dr. Bill Bass. He was the state forensic anthropologist at the time. He worked a case where he mistook a Civil War soldier for someone that had been dead about a year. It's a famous study if you ever read his book. So he was off by about 113 years in his postmortem interval investigation, and that was, to him, that was a reason to get this type of research story. So it took him to kind of spearhead this and start the facility and pursue it. And then I think once people realized that the contributions of doing experimental or generating experimental estimates, it took off from there. So it's been a very successful facility and program. What got me into it is pure accident. I am an environmental scientist and an ecologist by training. Never had interest in human bodies or medicine, did not want to go that route at all, and happened to take a visit to the anthropology facility in the first year that I was a faculty member. I was asked by one of the faculty there about decomposition. They were having some questions about how it occurred and microbial ecologist, what do you know about decomposition? And I said nothing, but I'll see what I can figure out. And in looking through the literature to try and answer their questions, I actually found that there was a dearth of studies on microbial communities associated with decomposition. We kind of knew the macroscopic changes and processes in decomposition, but we knew very little about microscopic events. And it just got me intrigued. I was hooked. I had a graduate student that was willing to work on this project, and that's how it all started. So very fortuitously, right place, right time.
So how is the necrobiome affected in a soil-free environment, such as a building or other non-interred environment?
A good question. And I don't know of any studies like this that are being carried out indoors. But it's certainly something that we should be looking at, particularly coming out of this forensic restoration conference. All their decomposition happens in an indoor environment. And so they were asking me the same questions. What's going to happen if you don't have soils? There are a couple good experiments out there where people have put cadavers or corpses onto sterile soils, and they find that they don't decompose at nearly the same rate as the ones. So we do need the environmental microbes to stimulate decomposition. So how much they're altered in an indoor environment, we're not sure. They have run decomposition studies in indoor versus outdoor environments and find generally that decomp is slower in indoor environments, and typically because it delays colonization by insects, and that's the primary reason so far. But we don't know much about the microbes and how they might differ.
How does time of year affect succession of the bacterial community? Do you see the same species coming in the same order, or do you see completely different species if something dies in the winter versus the summer?
We haven't done a seasonal study, but there are a couple other groups that have, and I think they do show that the microbial community composition does differ by season. So they do see different things in winter as opposed to summer. So part of that is probably seasonal things that are better competing under cold temperatures, and part of it may be the reduced insect load during the winter. So you don't have those insect communities coming in like you would in the summer. Good question.
Can you comment on the effects of altitude?
Oh. Good question. No, I'm not aware of any studies on altitude yet. This is a wide open area of research, so lots of room, lots of good questions.
Marissa Mendez and Matthew Harris from the YouTube chat are asking, what are the specific methods or specific sequencing techniques you can use to determine what genera of microbes are present, and how are soil microbes collected?
Okay, so we collect soil microbes using small cores, essentially just small tubes that allow us to collect things aseptically without introducing our own microbes into it. We can extract DNA from those soil cores, and we use a couple different approaches, but one of the main ways is to use a targeted metagenomic approach where we amplify a phylogenetically relevant marker gene. For bacteria, we use 16S, and we sequence all these 16S and compare it to known databases, and we can see what taxa are present. We've also played a little bit with shotgun metagenomics, which just sequence all the DNA in the system, and that gives you not just information on the bacterial taxa, but also fungal and other eukaryotic and insects. I mean, it gives you back all the DNA in the system, and so we've used that as well. And that has some advantages in that it tells you not just the types of microbes that are there, but the other things in their genomes, and so that can tell you what other functions they may be performing in the system.
So the University of Tennessee, you said it has like one of the first body farms. And now, are there other body farms across the United States or across the world? And how do, say, your baseline decay rates and stuff like that compare to other body farms across the world?
Yeah, so there's a couple other in the U.S., and I'm going to try and remember where they all are. There's one in Western North Carolina, there's one in San Marcos, Texas, there's one in Illinois, I think there's one in Colorado. Sorry if I'm forgetting your anthropology research facility. And so we do have a few facilities that are around the world, and we do have differences in decay between the facilities. So East Tennessee has hot, humid summers, and and Western North Carolina as well. So we probably have the fastest rates of decomposition. The anthropology research facility in Texas, I know, has much more mummification. Because they're a much drier climate, and so the tissues don't decompose as fast, and they dry out more quickly, and so you get a lot more dried tissue on the bodies compared to in the moist climate. We also have differences in scavenging between our facilities. So for some reason that I don't think anyone really understands, in Tennessee, we don't have issues with avian scavengers. We don't have vultures. I don't know if it's because it's all forested and they can't see it. I don't really know. But I do know that's a big issue in the Texas facility, and that they will get vultures coming to visit the bodies, and they frequently cage them to keep those scavengers out. So there's certainly climactic and even ecological differences in the different parts of the world. So I think that's why it's important to have these different facilities.
So Brian Rump from the YouTube chat asked, when does the thanatoscriptome leave off and the necrobiome take over?
So the thanatobiome refers to, I think, the immediate post-mortem time, and the necrobiome is kind of the outside. Is that what they mean? I've heard different definitions for the words, so.
Yeah, they said the thanatoscriptome.
Okay. I don't have a good answer for that. In fact, I don't even routinely use the word necrobiome, not that it's a bad word, I just say post-mortem microbes, and it sort of includes them all.
Have you looked at the differences in soil microbiomes between a place that has a constant influx of bodies, like the body farm, versus the surrounding woods or like a cultivated soil or anything like that?
Yeah, we do have some comparison sequence data on microbes at the Anthropology Research Facility in Tennessee and ones just outside, and there are differences. And we also notice differences in soil chemistry as well. And I think what's happening over the 36 years that this facility has been running, there's been such an enrichment with all these bodies that it's actually changed some of the soil chemistry.
And how have you been controlling for that?
Yeah, so we, in the studies that we're doing, we're lucky enough to get pieces of land that hadn't been used for bodies before. So the Anthropology Research Facility recently went through an expansion and got a new parcel of land. So we've been able to use fresh soils for that. And then bodies that maybe aren't directly being used in an experiment, but are being used to generate skeletons for their permanent collection, will go in the older part of the facility that's a little bit more impacted. We are actually playing around with ways of kind of bringing the soil back at the old facility, carbon amendments, and that sort of thing to see if that helps, adding mulch back in. Yeah.
Hi. The part of your talk where you mentioned the sort of dangers of pathogens from decomposing carcasses, earlier this year there was a pretty big outbreak of anthrax in Siberia, and they think it was caused by, due to the warmer climate, a reindeer carcass came uncovered from the permafrost, and essentially there was a huge outbreak of anthrax. And sort of thinking about, we have a bunch of pathogens that we may not be as concerned about anymore. They're eradicated or eliminated, and with decaying bodies and such, how much can you comment on what the concerns are? And even with like a changing climate, where do you predict that there would be vast differences in your work?
I wasn't aware of that, and that sounds like a very interesting and scary finding. Yeah, we were certainly surprised by our findings. We really didn't expect some of these, in particular, obligate anaerobes. We really didn't think that they should last that long in this environment. We didn't think that they would be much of a risk. And they did, and so that does speak to the possibility that you could have events like this where an animal that dies or a human that dies from some disease still carries that pathogen, and if it's preserved in some way, may come back to life. I don't know, I don't really want to be a fear monger and say that this is going to happen all over, but I guess the possibility exists, especially if we have reindeer carrying anthrax. That's scary. Yeah.
It's a little different for it, but the fact that you have persistent anaerobes for such a long time is interesting.
Mm-hmm. I will say, we don't know if the anaerobes are active, right? So our techniques are using DNA signatures to look for these organisms, so we are getting their DNA out. That doesn't mean that they're alive or that they're doing anything. That's still to come, so.
All right, so we're going to have one more question, and then Jennifer will be at the reception, so you can talk to her there.
Sure, why not? Okay. I was wondering whether or not your lab is thinking about looking at other variables like age or gender and how those bacterial communities differ as the body is decomposing, because we know that, you know, younger people or older people are generally colonized by different things, and you do see differences between men and women too. So I was just wondering, do you see that, or is it, by the time someone is already decomposing, is it just like a set group of bacterial communities?
It's a good question, and we have almost no data on young people and decomposition because we don't get a lot of donors. Just the nature of the program is that we tend to get a lot of elderly people. And in the case where young people die, it's almost never natural, and so they're almost always autopsied. And if they're autopsied, we can't use them for decomposition studies because it wildly changes decomposition dynamics. So we haven't been able to experiment with young people, but it's a good idea.
All right, let's give Jennifer a hand for a great talk. And thank you to all of you who came in person and everyone who watched online. If you're here in person, we now have a reception out those doors. So please enjoy yourself.
They tell you to put your chin right there.
I became an ASM member in 1981.
95?
Since 2008.
I joined ASM in 1974.
I've been an ASM member for 41 years.
I first became a member in 1989.
Seems like all my career.
I'm willing to bet it's a decade.
My first ASM was 1993. I still have a picture of me holding the program open, sitting on the hotel room bed, really excited because for the first time ever I saw my name in print associated with microbiology.
When I was a graduate student at Purdue in 1980, it was the thing. I mean, if you were going to be a microbiologist, you joined the ASM. It was just really that simple.
ASM is very special to me because I became a member in 2002, and when I first attended the meeting at Salt Lake City, Utah, I met my PhD thesis mentor, Dr. Arturo Casadevall. And so ASM kind of introduced me to the PhD world.
Definitely the most important thing is that ASM has provided me with a graduate fellowship. So they've helped support me during my graduate training. In addition, I've gotten a lot of networking opportunities and I've met a lot of really great people through ASM.
And we can share our work and connect, and they can teach me things that I don't know and I can teach them things that they don't know. And just that partnership and working together, that's what scientists do, to share your work. And it's the most exciting thing to learn about new projects.
ASM has actually done a whole lot of work and given me the kind of exposure I wouldn't have had if I'm not a member of ASM.
Many brains are better than one.
Great thing. It's really member-driven organization. I love that. I've always loved that.
Could make the bigger the better, the more the merrier.
You want to do microbiology, become a member.